Welcome to class number 2, session number 3. New bottom-up techniques are being explored as a complement to traditional top-down methods. In contrast to removing the materials we don't need, bottom-up techniques simply construct the desired features from fundamental building blocks, usually throughout self-assembly, without the need for patterning. Indeed, in the bottom-up approach, materials and devices are built from molecular components which assemble themselves chemically by principles of molecular recognition. In molecular recognition, molecules can be designed so that specific configuration or arrangement is favored due to non-covalent intermolecular forces. Thus, two or more components can be designed to be complementary and mutually attractive so that they make a more complex and usable as a whole component. Bottom-up approaches should be capable of producing devices in parallel and be much cheaper than the top-down methods, but could be potentially be overwhelmed as the size and complexity of the desired assembly increases. Most useful structures require complex and thermodynamically unlikely arrangements of atoms. Nevertheless, there are many examples of self-assembly based on molecular recognition in biology, most notably the Watson-Crick base repairing and the enzyme-substrate interactions. The challenge for nanotechnology is whether these principles can be used to engineer new constructs in addition to natural ones, growing these structures in well-designed coordination and fabricating robust structures, etc., etc. A key approach in the bottom-up fabrication technique is the self-assembly. Self-assembly in the classic sense can be defined as the spontaneous and reversible organization of molecular units into ordered structures by the non-covalent interactions. The first property of a self-assembled system that this definition suggests is the spontaneity of the self-assembly process. Indeed, the interactions responsible for the formation of the self-assembled system act on a strictly local level. In other words, nanostructure builds itself. There are at least 3 distinctive features that make self-assembly a distinct concept. The first feature is the order. The self-assembled structure must have a higher order than the isolated components, be it a shape or a particular task that the self-assembled entity may perform. The second feature is the interactions. Self-assembled structure rely on slack interactions such as van der Waals, capillary, and hydrogen bonds. Of course, this is given with respect to the traditional covalent, ionic, or metallic bonds. Although typically less energetic by a factor of 10, these weak interactions play an important role in material synthesis. It can be instructive to note how slack interactions hold a prominent place in materials, but especially in biological systems, although they are often considered marginally with respect to strong, namely the covalent interactions. The third feature is the building blocks. The building blocks are not only atoms and molecules, but span a wide range of nano and mesoscopic structures with different chemical compositions, shapes, and functionalities. These nanoscale building blocks can in turn be synthesized throughout conventional chemical routes or by other self-assembly strategies. There are 2 types of self-assembly: intramolecular self-assembly and intermolecular self-assembly. The intramolecular self-assembly molecules are often complex polymers with the ability to assemble from a random coil conformation into well-defined stable structure. An example of intramolecular self-assembly is a protein folding. On the other hand, the intermolecular self-assembly is the ability of molecules to form supramolecular assemblies in the sample. A simple example of the formation of a micelle by surfactant molecules in solution is one of the intramolecular examples. Following is a short video that demonstrates the self-assembly of a dendrimer host gas system along with the animated presentation. In the research shown in the animation, researchers make use of supramolecular or non-covalent chemistry to assemble nanoparticles with small hydrophobic patches. The number of patches depends on the coverage of hydrophilic guest molecules, shown in blue, that protects the hydrophobic host molecules, shown in yellow. The coverage, in turn, depends on the concentration of the guest and host molecules on solution. In most systems, assembly of nanoparticles into large structures occur at high concentration, and dilution causes them to be disrupted. Dilution of a solution of host and guest complexes leads to an increased number of attractive hydrophobic patches, which interact and drive assembly. In other words, this system assembles upon dilution, seemingly defining the law of mass action. We predict that this study could help close the gap between complex biological and colloidal-like systems. Self-assembly can occur spontaneously in nature, for example in cells and other biological systems, as well as in human-engineered systems. It usually results in the increase in internal organization of the system. Many biological systems use self-assembly to assemble various molecules and structures. Imitating these strategies and creating novel molecules with the ability to self-assemble into supramolecular assemblies is an important technique in nanotechnology. In self-assembly, the final or the desired structure is encoded in the shape and the properties of the molecules that are used, as compared, of course, to the traditional techniques such as lithography, where the desired finite structure must be carved out from a larger block of matter. An example of biology-based self-assembly process, I will give the example by a scientist at the United States Department of Energy, the DOE, at Brookhaven National Laboratory, who reported successful assembly of three-dimensional multi-component nanoscale structures with tunable optical properties that incorporate light absorption and emitting particles. In this work, the scientists used DNA linkers with free binding sites to connect gold nanoparticles and fluorescent dye molecules tagged with the complementary DNA sequences. The DNA linker molecules had three binding sites. The two ends of the strands were designed to bind to complementary strands on plasmonic gold nanoparticles, namely particles in which a particular wavelength of light induces a collective oscillation of conductive electrons, leading to strong absorption of light at that wavelength. The internal part of each DNA was encoded to recognize a complementary strand chemically bound to a fluorescent dye molecule. This setup resulted in a self-assembly of three-dimensional body-centered cubic crystalline structure with gold nanoparticles located at each corner of the cube and in the center, with dye molecules at defined position in between them. Following is a short video that demonstrates the self-assembly of binary virus gold nanoparticle superlattices along with the animated presentation. Indeed, in this animation, the Aalto University-led research group shows that the CCMV virus or ferritin protein cages can be used to guide the assembly of RNA molecules or iron oxide nanoparticles into three-dimensional binary superlattices. The lattices, which will be seen soon on the screen, are formed throughout tunable electrostatic interaction with the charged gold nanoparticles. An important bottom-up aspect for sensing applications is the so-called self-assembled monolayers. Self-assembled monolayers, or usually abbreviated as SAM, S-A-M, of organic molecules are molecular assemblies formed spontaneously on surfaces by adsorption and are organized into more or less large ordered domains. In some cases, molecules that form a monolayer don't interact strongly with the substrate. On the other hand, and in other cases, the molecules possess a functional group that has a strong affinity to the substrate and anchors the molecules to it. Such a self-assembly monolayer consisting of a head group, tail and functional end group is depicted in the figure presented on the screen. Common head groups include thiols, silanes, phosphonates, etc., etc. Self-assembled monolayers are created by the consorption of head groups onto a substrate from either the vapor or liquid phase, followed by a low organization of tail groups. Initially, at small molecular density on the surface, adsorbed molecules form either a disordered mass of molecules or form an ordered two-dimensional lying down phase, and at higher molecular coverage over a period of minutes to hours begin to form three-dimensional crystalline or semi-crystalline structures on the substrate surface. The head groups assemble together on the substrate, while the tail groups assemble far from the substrates. Areas of close-packed molecules nucleate and grow until the surface of the substrate is covered by a single monolayer. Thin-film self-assembled monolayers can be placed on nanostructures. In this way, they functionalize the nanostructure. This is an advantage because the nanostructure can now selectively attach itself to other molecules or self-assembled monolayers. This technique is useful in biosensors or other devices that need to separate one type of molecules from its environment, as we will learn in the next classes. After the introduction of the various top-down and bottom-up fabrication approaches, I will present a video from the Johns Hopkins University that demonstrates devices fabricated by a combination between these two approaches, namely the top-down and the bottom-up or the self-assembly approach. Indeed, the following video presents the production of three-dimensional structures by the assembly of patterns made by photolithography. Enjoy.
Once the two-dimensional structures have been prepared, they are heated until the hinges begin to melt. As the hinges melt, they ball up in order to minimize their surface area, and since they are attached to the square faces, this brings the faces together and eventually forms a cube.
This project demonstrates the use of these polyhedral containers for chemical encapsulation, remotely guided chemical release, and spatially controlled chemical reaction. This project demonstrates the hierarchical self-assembly of complex polyhedral microcontainers, such as the dodecahedron shown here. Polyhedral microcontainers that approach a sphere are especially attractive for biomedical applications, such as drug delivery in the body. Shown here are the two-dimensional templates prior to being self-assembled. Each face of the container can have unique surface patterning, and bead encapsulation within the dodecahedral containers has been demonstrated. This project demonstrates an alternative to surface tension-driven self-folding. This new strategy utilizes thin film hinges that allow self-assembly to be triggered on demand in water, using relatively low temperatures. This project demonstrates mass-producible micro-grippers that can be remotely triggered by temperature or chemicals under biological conditions. These microgrippers utilize the previously mentioned thin film hinges. To demonstrate controlled remote movement, two magnets were used to guide a microgripper from one opening of a spiral tube to the other. This movie shows a microgripper being remotely moved towards a blue bead using a magnetic stylus. Once over the bead, the microgripper is triggered to close by slightly heating the solution. After capture, the microgripper with the bead is moved away using the magnet. This movie demonstrates the remote manipulation of a microgripper into a capillary tube and remote retrieval of cell mass that is dyed with red stain. The movie highlights the applicability of the microgripper to capture living cells, such as in a biopsy. A hypothetical future biomedical application for these microgrippers is controlled movement throughout the body to carry out non-invasive procedures, such as a biopsy. This project demonstrates microgrippers that can be remotely opened and closed by chemical cues. These microgrippers can effectively move objects from one location to another. The last project that we'll discuss demonstrates a new strategy to construct complex 3D pattern structures by self-assembling 2D sheets that have thin film hinges. As an example application, these sheets can be used as microwells for cell culture.
With this video, we come now to the end of class number 2, session number 3. Thank you.